I'm Mark Keel, founder and chief science officer of Genomic, where I supervise our data licensing to pharma. Today I want to introduce to you the concept of a comprehensive genomic landscape and describe through examples how it is useful in the context of rare disease. The outline of my talk is to first describe why using genomics in rare disease can provide benefit and what are some of the applications of genomics in the context of rare disease. Secondly, to describe how one goes about gathering the needed evidence to culminate in what we've referred to as a comprehensive genomic landscape. And then lastly, and in the meat of my talk, to describe through examples what some of the representative use cases are of comprehensive genomic landscapes in a variety of different rare diseases. Let's start by talking about the core benefits and applications of genomics. By now, it's well known to most of you that drug targets with genetic evidence are associated with an increased likelihood of successful development. In fact, from a study by Matt Nelson at GSK some years ago, a retrospective study found that there's a twofold increase in success among drugs that have genetic evidence supporting their candidacy. Given that 80% of rare diseases are genetically driven, finding this genetic evidence becomes crucial. In particular, genomics can clarify the cause of a complex disease, possibly clarifying similarities across multiple related diseases, or otherwise resolving pathway heterogeneity or uncovering latent homogeneity within disease pathways, and importantly, providing robust and impactful data where little background information may be available, as is typically the case by definition for most rare diseases. Specifically, in the context of rare disease, genomics can empower pharma to optimize identification of preclinical targets, speed downstream R&D and reduce overall costs, maximize the success of a clinical trial, expedite FDA approval, all benefits that I'll be illustrating in use case examples through Genomic's client work with various pharma groups. What's required to get to this evidence? Specifically, a comprehensive understanding of genomic biomarkers and how they inform diagnosis, how they are associated with functional effects on drug pathways, and what evidence may exist in support of their actionability is what we're talking about when we speak to genomic evidence in support of a drug candidate. How are we going to get there? What's required to assemble a comprehensive genomic landscape to achieve this insight? A comprehensive genomic landscape requires identifying and interpreting all of the genes and variants that are associated with a disease, and how there is any therapeutic information that pre-exists in the empirical evidence to support hypotheses for a candidate that you're pursuing. The questions that need to be addressed are: what are the genetic drivers of disease? Which variants have clinical evidence to support their pathogenicity? What are some functional studies that may describe the mechanism of action or the otherwise describe biologically the way that the variant contributes to disease? What therapies have been tested and found to be efficacious or have not been found to be efficacious? And critically, what's the supporting evidence to address any one of these questions? What I want to impress upon you is that the answers to these questions are contained within the many decades of empirical evidence, whether they be from decades ago or from as recently as last week. Assembling and interpreting this evidence is what will lead to a twofold increase in success rate of your drug target with that evidence. However, manually extracting this information is extremely challenging, very time-consuming, highly error-prone, and usually results in a highly incomplete data set that lacks the sensitivity required to fully realize the potential of genomics in rare disease. At Genomic, we've devised a method to extract this information from the literature and to organize it into what we've referred to as genomic associations. Specifically, Genomic can identify and connect genomic concepts in the form of genes and their variants, and how they interact with and are associated with diseases and phenotypes and any tested therapies in the clinic or in research, And then to better annotate that information with a whole battery of categorized key terms, be they functional or clinical, as befits a specific disease, gene, therapy, or phenotype. Then over on the right, what I'm illustrating is having the benefit of a comprehensive view of the empirical evidence to support these genomic landscapes can provide a great deal of ballast for and supporting evidence corroborating your assertion for the genomic association being useful in your pursuit of a particular drug candidate. Specifically, evidence in support of a gene being associated with the disease, as well as being associated with an efficacious therapy, will provide a great deal of scientific support for your hypotheses as you're pursuing a drug target. Assembling all of this information and interpreting it culminates in what we've referred to as a comprehensive genomic landscape, which is to say a complete data set of all variants in any gene or gene pathway, all expertly curated according to clinical standards, ACMG for constitutional and AMP for somatic disease, with fully referenced, evidence-cited insight from clinical studies and their findings, as well as in vivo or in vitro empirical functional studies, and all necessary associated treatment and trial information with evidence citations from the primary source of that information, which is to say the medical literature and ancillary databases. All of which is summarized for efficient reporting and immediate actionability, and is kept up to date on a quarterly basis with any new and relevant findings. At Genomic, we've performed these comprehensive genomic landscapes for more than a dozen large pharma and biotech companies. And where I really want to go with this talk is a description of some of those case studies from our engagements with those pharma groups in the context of rare and genetic disease. The way that I've articulated these use cases is from a benefits perspective, categorizing them into preclinical as well as clinical and post-commercial activities. The first examples that I'll highlight involve optimizing research and discovery activities. I'll begin with an example that speaks to using a comprehensive genomic landscape to identify and assess a new target, and then to showcase how genomic landscapes can be useful to provide insight into the mechanism of action of a drug and the pathogenicity of variants in the context of a protein as it's associated with a rare disease. And then the latter examples that I'll showcase will be more on the clinical side, where I'll begin by talking about how genomic landscapes can inform clinical trial candidacy for individual patients, or can otherwise be useful to identify patients at point of diagnosis. And finally, talk to how comprehensive genomic landscapes can be used to assemble a companion diagnostic and useful to assemble all of the evidence necessary to produce a cogent regulatory submission to expedite, say, FDA approval. The first example I'm going to describe involves assessment of a new target. In each of these examples, I'll begin by describing very briefly the gene involved, though it may just be representative, as well as the disease of interest, and speak to what the baseline understanding of the group that we were doing this work with was at the time that we began our project together. I'll then talk about what the culmination of our comprehensive genomic landscape was and how that comprehensive genomic landscape brought value to the client that we were working with. In this case, for new target assessment, I'm going to be speaking to the comprehensive genomic landscape for TARD-BP in the context of amyotrophic lateral sclerosis, or ALS, which is a neurodegenerative disease, which is in fact a rare genetic disease. The baseline understanding was 20 disease-causing variants that were cultivated from a review of data provided by ClinVar. The culmination of the production of a comprehensive genomic landscape by Genomic resulted in a nearly 5-fold increase in variants in this gene that had demonstrably pathogenic evidence to support their clinical utility. So the takeaway from this study is that ClinVar does not provide the whole picture. So reliance on ClinVar solely means that you're necessarily missing valuable information. To illustrate this, I've depicted each of those variants in gray from ClinVar at the top and in blue from Genominon's comprehensive genomic landscape at the bottom, where you can see each one of those individual dots reflects a variant and all of the evidence underpinning it for that clinical assessment. They're arrayed along the linear axis of the protein with a reflection of the functional domains of that protein. And as those dots stack up on each other, that's an illustration of how each of those individual variants cluster within the protein across multiple patients and functional studies. In this example, you can quite clearly see that there's a dramatic increase in the number of variants that Genominon's comprehensive genomic landscape uncovered. But if you look closer at the data, you see that only with the full view of all of this evidence does a pattern emerge that was not otherwise apparent from the ClinVar data alone. Specifically, in the case of TARD-BP, there's a whole category of variants in the RRM domains of TARD-BP that were not present in ClinVar, where the evidence from our comprehensive genomic landscape indicates that those variants are found in patients, as well as corroborating functional evidence that indicates that the function of that protein is abrogated in the presence of those variants. So there's a great deal of evidence to support the benefit of knowing those variants if you're to fully understand how TARD-BP is associated with ALS. The next example that I'm going to speak to has to do with divining the mechanism of action of mutations in the GBA protein in the context of Parkinson's disease and Gaucher's disease. The former being a neurodegenerative disease and the latter being a metabolic disorder of childhood that is properly a rare disease. In this case, the goal here was to reconcile the way that variants in the GBA protein can lead to both Parkinson's and Gaucher's type 1, which both have neurophenotypes, but have no relationship to Gaucher's type 2 and type 3 from a central nervous system perspective. The baseline understanding that our customer began with was about 36 variants that they had cultivated over about 2 years of manual investigation of the literature. When Genominon produced its comprehensive genomic landscape, we increased by 8.5-fold the number of demonstrably disease-causing variants, each with supporting evidence, including 114 of which had functional evidence from in vivo or in vitro studies. And we assembled this data all within a matter of weeks. The net result here, the value that we provided, was a unique insight into disease development for Parkinson's and Gaucher's disease. Just to showcase what that swarm plot looks like, again, each of those dots is a unique variant with the necessary evidence from the medical literature to support its pathogenicity, arrayed along the linear axis of the protein to showcase where there's recurrent regions of the protein that are mutated in this disease context. This is all of the variants that were found to be pathogenic from that study for Parkinson's and for Gaucher's disease. But then in the process of assembling and curating this data, we more finely parsed that evidence, breaking it out according to each of those individual diseases, which I'm showcasing on multiple rows in this compound swarm plot. At bottom in gray are the variants that we found were associated with Parkinson's, and then immediately above that in navy are the variants that were found to be associated with Gaucher's. And then in the 3 upper rows, the swarm plots depict how those variants found in Gaucher's break out according to specific disease subtypes. So when I highlight these regions, this is a reflection of how Gaucher's type 1, which has a neurophenotype, and Gaucher's type 2 and type 3, which do not, how the variants in Gaucher's type 1 have regional affinity with the variants found in Parkinson's disease. So without going into detail about the mechanism of action, the comprehensive genomic landscape in this case gave a great line of sight into why some variants in GBA cause types 2 and type 3, and others cause Gaucher's type 1, and what that may mean in the context of the phenotypic and clinical relationship between Gaucher's type 1 and Parkinson's disease. Another example of the benefit of comprehensive genomic landscapes in the context of rare disease is involving patient identification. In this example, we were looking at the ATP7B gene, which is associated with Wilson's disease. And the baseline understanding, again, in this case, was from ClinVar, where about 250 variants were found to be pathogenic based on clinical information from user submissions. And when Genomenon put its attention to the comprehensive genomic landscape for ATP7B, we increased by 2.5-fold the number of pathogenic variants from 248 to 631, including a full 145 of those variants with corroborating functional evidence. So overall, this was a dramatic increase in diagnostic yield for Wilson's disease, which has a complicated genotype-phenotype correlation. The evidence for strength of genotype-phenotype correlation all was patterned in the exhaustive annotation and reference citation of our comprehensive genomic landscape. Just to visualize the difference between what ClinVar provided at the top in gray and what resulted from a truly comprehensive genomic landscape from Genomenon's work in the bottom in blue, this is to illustrate how there's dramatically more variants that are available to you for diagnostic purposes to increase the number of candidates that you can enroll with objective criteria, such as a genomic assay, say, at the onset of your clinical trial. Another example of the benefit of comprehensive genomic landscapes pertains to how patients are enrolled in clinical trials. So this is, again, from a case study from an actual pharma client of ours. We've done webinars with this group in the past, and we have an ongoing relationship with them. Their interest was in monogenic obesity and the genes that are associated with monogenic obesity, including the LEPR gene, among others. Their baseline understanding, culminating from many man-years of manual review of the literature, was in clarification for 3 of the genes and 12 of the variants within those genes that they found to be demonstrably associated with monogenic obesity. When we turned our attention to this whole pathway of genes, we found that there were between 40 and 120 genes, 48 of which we have fully curated, resulting in 660 variants with that clinical grade demonstrable pathogenicity based on clinical and functional evidence from the medical literature. So overall, this resulted in a 55-fold increase in the number of variants that were demonstrably disease-causing in the context of monogenic obesity. And for this particular group, resulted in a fundamental change in their clinical trial enrollment strategy, whereas instead of being a static enrollment strategy, it has now evolved into a rolling clinical trial admission. Just to illustrate in a different way, with a different swarm plot, the value of the comprehensive genomic landscapes that we're discussing here, in red, open and closed circles, I'm highlighting those variants that are both pathogenic and likely pathogenic, which are functional equivalents. But then also in gray, what I'm highlighting are those variants with an uncertain significance designation, though, which have either functional and or clinical evidence that they may be pathogenic, that they're just waiting on new information to be published in the medical literature, or otherwise assembled by the pharma company themselves to take that evidence over the threshold from being uncertain to being in fact likely pathogenic or pathogenic. So this is illustrating, again, how the comprehensive genomic landscapes that we're discussing have as an evidence predicate a rigorous framework around which to assess the pathogenicity of each variance, and that there's a great groundswell of information in the rest of that comprehensive genomic landscape that has some sub-threshold amount of evidence that with any new paper that's published may go over that threshold, illustrating the benefit of keeping apprised of newly published information in this way. Another example has to do with the benefit of using comprehensive genomic landscapes in the context of a regulatory submission, and in particular, producing a companion diagnostic. This example centers around the RET gene in the context of multiple endocrine neoplasia syndrome type 2, or MEN2. The goal for this project was to understand all of the causative variants in the RET gene, and the baseline that we began our understanding with here with this client was 38 variants that were known based on manual review of the literature to be disease-causing. And our comprehensive genomic landscape process culminated in fully 130 variants, each of which was activating in the context of RET, having both gain and loss of function mechanisms, where only in the case of MEN2, activating mechanisms of action were relevant for the therapy. This resulted in a 3.4-fold increase in the number of known variants that had demonstrable pathogenicity based on the evidence, which I'm illustrating here on this swarm plot, where the top is that baseline understanding of the variants that they understood from their literature review. And at bottom was what Genomic uncovered in what is now a truly comprehensive genomic landscape. And so in addition to better understanding each of the unique variants at a given hotspot region, as you can see at the tallest peak there in the center, having the benefit of a comprehensive landscape, as you see depicted here, uncovers patterns that would otherwise not be apparent if you only had a fraction of the data, as was the case when we began this study with this client. So you're seeing sort of satellite hotspot regions for which there was next to no or no information available previously, that only emerged with an exhaustive curation and cataloging of evidence from the medical literature. Hopefully with that introduction, the description of what a comprehensive genomic landscape was, and some illustrative use case examples with a window into some of the findings that we were able to provide for our clients to give them greater insight into their drug development pipeline, you have a greater appreciation for the benefit of genomics in the context of rare disease, and particularly how comprehensive genomic landscapes can increase the likelihood of success of your drug development program.